So glad to have you here. I'm Jane Dutton, and I'm one of the members of the Adult Faith Formation Committee, and we have a wonderful season and a wonderful session today. We're very happy to have Dr. Bill Gruber share an amazing story about the development of the COVID vaccine. Bill is right now the principal of Gruber Vaccine and R&D Consulting. He's the former Senior Vice President and Head of Vaccine Clinical Development and Research and Development at Pfizer. He retired in 2023, and then he and his family moved here to Ann Arbor, so they're relative newcomers, and it's so wonderful to have them. He has 4 years of experience in vaccine research and development, and in addition to his knowledge in School of Medicine at U of M, and also adjunct clinical health. Today he's going to share with us a story at the intersection of science, medicine, and faith as applied to people working on the COVID vaccine. His talk, he says, is aimed to be a tribute to the people Bill worked with to make the vaccine happen. And I just have to tell you, one fun common link is that our grandchildren are in the same classroom at Arbor White School. So Bill, thank you so much for coming. We really appreciate it.
Well, again, thank you, Jane, very much for that introduction. And I want to thank Mel. She was kind enough to and certainly Brent Ivy and Bill Hall in the back. I'm going to probably abbreviate some sections because I really enjoyed the service, but it ran a bit long. But I'm hopeful that you'll get the main message that I hope to deliver today. And if I'm successful, you'll have a better understanding of what actually happened with the COVID-19 vaccine development. And most importantly for me, and new for me, I've never had a chance to really talk about the people that made this happen, who come from a variety of backgrounds, a variety of faith journeys. And then it's really up to you, as you hear this story and how the pieces came together, to see what you think about God's hand in all of this. And I think what better day than Communion Sunday Day, and with some of the messaging that we heard this morning, to hear about that message. So here, I really like this definition of providence, and I'd like for you to keep this in mind as we talk about this. God conceived as the power sustaining and guiding human destiny. And I think if you're like me, you'll see God's hand in what I'm about to tell you. So you've already heard enough about me, so I'm going to dispense with this in terms of my secular bio. But I do want to share with you a little bit about my faith journey, and then more importantly, how faith and the various backgrounds of other people have contributed to making a COVID-19 vaccine. So first of all, as I said, this is a bit of a confession. I had a very casual relationship to the church in my youth, no real use for Sunday school. I was baptized at age 6, confirmed in middle school. I asked nerdy questions of the pastor at that time, Charlie Shedd, in adolescent gatherings, largely driven by my interest in physics and sort of blending that with Christianity. And so I'd ask questions like, well, if Christ came to save us on earth, if there's intelligent life elsewhere in the universe, is God sending Christ everywhere? And those sorts of questions. And I had sort of this view that I didn't know this notation at the time, but this is kind of the view that I held from Ernest Rutherford, that all science is either physics or stamp collecting. It served him right, by the way, that he won the Nobel Prize not in physics but in chemistry. I largely reconciled my thinking that physics might explain the what and the how, but not the why. And that's where faith, in my view, comes in, is better understanding where do we stand in relationship to God. And I like to think about where we are now. We're learning more and more about the what. Some of you may recognize this is my favorite picture from the Webb telescope, and you may have heard this description. But if you were to basically hold up a grain of sand, and that represents the part of the sky that you blot out at arm's length, this is what you would see looking 4.6 billion years into space-time. So that tiny fraction has tens of thousands of galaxies. So the universe is more vast than we can comprehend. But what I like to think is, as vast as the universe is, this is what God thinks about, and this is from a New Yorker cartoon, which is one of my favorite cartoons. The Milky Way detail. He cares about us. And so in that context, although we are not the physical center of anything, we are the center of God's love. We are children of God. We're not the center of the solar system. We're kind of in a suburb of the Milky Way. We're way off out in the distance in terms of the universe. And I like to think about this in the context of Jesus. Who did Jesus serve? He served the marginalized, the outcast, the sinner. Well, it's hard to be, at least metaphorically, more marginalized than out as a speck somewhere out in the universe. So to me, that's a remarkable way I like to think about this. And so given that, throughout life, and obviously I hopefully have matured in thinking about this over time, I prayed with the belief that God expected something of me, but what was that going to be? And my mother was often fond of quoting this verse. The context is a little challenging, as I expect Nathaniel and Tom will know, but the message of this particular verse, I think, is important. For everyone to whom much is given, and my mom constantly reminding me of how much I was given, from him much will be required, and to whom much has been committed, of him they will ask the more. Now, in thinking about this, one of the pivotal moments in my life occurred when I was in high school, and I was handed 2 pieces of wood like this, and I have these obviously to this day, to think of the cross as an eye with the minus mark through it. And so the notion is that we're not here to serve ourselves, but to serve God. And this has stood with me ever since. And of course, my family will tell you I don't always live up to that. In fact, probably less often than more often. But I think that's a critical ingredient as we think about faith, to think about the cross in so many ways, but in the way of an eye with a minus mark. And then, fortunately for me, and I don't know that this exists in the Presbyterian Church USA anymore, there was a course that I had to take, sponsored by the church, perhaps related to predestination, perhaps related to calling, in which I met with a mentor who walked me through a booklet as I explored my faith and explored what was of interest to me, what should I do, and in fact it ended with courses. And it was at that point I began to pivot and think, well, maybe it's not physics that I'm really supposed to be focused on. Maybe it's more medicine and biology. All the things were sort of pointing, including the test, and said, you're not going to be so happy as a physicist, but you might be happier as someone who works in medicine and biology. So that was important. But I think this is something that probably resonates with all of us. Another quotation that I think is really important, and it hits me on more than one level, and that is the quotation from Mark 9:24. Lord, I believe, help my unbelief. I think if you're like me, there's always the tension between faith and doubt. I think Paul Tillich pointed out that faith is actually doubt is actually an important part of faith because it causes you to probe further, to explore further. I've taken a great deal of comfort from that. The other reason that this strikes me is I'm trained as a pediatrician. And if you know this story, the story is one where the father who makes this statement comes bringing his son, who for all intents and purposes, as I've looked at Scripture, had a generalized seizure disorder, and basically Christ healed. And so, again, just resonant for me as a pediatrician and as a person of faith. So I had a lot of help, by the way, along the way in terms of helping me gain in my faith. First at Memorial Drive Presbyterian Church, then Westminster Presbyterian Church, which has a connection with Mary Elizabeth because that's where Pastor Toomey was, and he ultimately, I think, taught some of her courses in seminary. And then First Presbyterian Church in Goshen, and throughout this path, that was a more light church, meaning a more expansive view of God's love for everybody, regardless of race, ethnicity, sexual orientation. But it wasn't obviously the buildings, it was the people that were there. And what I really wanted to emphasize as I go through this throughout, that it's the people that made the difference, and the intersection of people across the same sort of diversity I was just talking about, the same diversity that brings forth God's love. So, no one more important than my wife, Gina. Here we are on our honeymoon. We were already sort of contrarians in a sense. I mean, after all, who goes to Vail, Colorado to cross-country ski, but that's what I do. You can see those stylish pants that I'm wearing. And this is a circumstance where there's a little bit of an intersection with Pastor David's history. My wife went to Baylor College, well, she did go to Baylor College of Medicine, but she first went to Baylor University in Waco, that we lovingly referred to as the Baptist Vatican, in the period that I would refer to as BCJE, which just stands for before Chip and Joanna Gaines, basically gained importance in that area. And yet, we did have a chance to go back, and here we are just a few years ago at Magnolia Hill. But Gina was far ahead of me in so many respects. First of all, as a high school student, she was going down to Nicaragua delivering immunizations. She secondly taught English as a second language while she was in college. And he was the person who really brought me back to the church. Trained or raised as a Baptist. Her father was a lay Baptist minister. She had me coming to the church when I'd go to visit Waco. And to be honest, for all of you that give sermons and you know, disparagement, I can't remember a single sermon. But what I can remember is this one vivid imagery. I know that David and all our ministers like telling stories about a story about a pastor who had a whole group of students, and his main message would be that God is love, as he would thump each of them on the head with the Bible. And that stuck with me forever. So obviously foundational to my entire being is my growing family, and represented here is John seated, Gina, my son Joseph, Thomas, yours truly, my son Paul, Bridget, Sam and Ellen, and they're the main reasons that we actually have moved to Ann Arbor, is to be closer to them. They keep me honest. They no doubt will have comments once they see this streaming or otherwise about how I could have done it better. Feel free to offer up your own. But let me tell you about this person. Talk about predestination, which Presbyterians obviously like to talk some about. How could Chapel Temple become anything but a clergyman? And in fact, he did. But I knew him before he did such remarkable things that served as an example for me, like smuggling Bibles during college, 2 cells. This was again back in the 1970s when the Iron Curtain was still present. He was smuggling Bibles and literature to Christian cells over the summers. He and his wife were the first house parents for the Ronald McDonald House that was newly built in Houston for the surrounding hospitals, while he was getting his PhD at Rice University in religion, and while he was pastoring a church. So I felt like, gosh, I was busy, but I couldn't hold a candle to him. He has been a mainstay for me in terms of my journey. He's been encouraging throughout my wife and my life. So, and the main reason, the way I meant to tell you, the way that I met him was in the 6th grade. So it's been since the 6th grade when he beat the pants off me in a creative writing contest, and we became friends ever since. All right, but the real story as far as COVID-19 vaccine begins with something represented in this picture. So that hairy-looking nerdy guy on your left is me. The guy on the right is Barney Graham from the small town of Paola, Kansas, high school graduating class of 60. He worked on a hog farm. We were thrown together. Some might argue that this was just random. Again, you may see something other than that as we go through the rest of the story. We had no idea at the time that our lives would continue to intersect, and in fact, he'll be my house guest later this week. So how did we end up intersecting? It came to be that we began to work in our careers on respiratory syncytial virus. This was an important virus because it's the leading cause of global infant respiratory disease. Infection leads to a large number of deaths throughout the world. It's the single most common reason for hospitalization of children in the United States, 40,000 to 120,000 every year. And so behind the scenes, while we were still in college, there had been an attempt to develop a vaccine, which is a formalin inactivated RSV vaccine that was disastrous in terms of its outcome. And you see that represented here. When you look in the middle of the page there, you see formalin inactivated RSV. Number of hospitalized individuals when they got RSV was 80%. when the expectation in the control arm showing there as FIPIV1 was 5%. So not only did it not protect, it made the disease worse. It was a catastrophic event. And so off I go to Baylor College of Medicine across the street. Barney goes to KU. And these people prove fundamental in terms of my further education. And in the interest of time, and I'm not going to talk a lot about this here, but I'm happy to answer questions about each of them. I'll just say that Carol Baker has spent a lifelong quest for working on B strep. She was first my teacher, mentor, colleague, friend, a woman of strong Christian faith. Ralph Feigen, the most charismatic individual I ever knew, who would do such things as on rounds would pick somebody out who's athletic, like you look pretty athletic. He'd say, I bet I can beat you up the stairs, and would run up the stairs in the middle of rounds and race people. It was really phenomenal. And he brought along Shelley Kaplan, who was the most practical person in understanding how to take care of people well, both in terms of their mental and physical well-being. So how could I not go into pediatric infectious disease? These people all arrived on the scene at the time I was doing my pediatric rotation, and it inspired me ever since. But this person in particular, so I still had to think, well, gee, I'm inspired about infectious disease. Where is my niche? And it was at this point that I began to appreciate the story that I just told you about RSV and the inability to have developed a vaccine successfully. This guy, Paul Gleason, said, well, gee, we know that not every woman who gives birth to a child ends up having a child who ends up in the hospital to RSV, yet all of them, by the time they're 2 years of age, will be infected. So what's going on? And his point was that, in fact, antibody, mother's gift to the baby, protected those kids and kept them from going into the hospital. And so how did I get involved with this? I was actually collecting specimens and identifying children from whom he had the antibody samples to demonstrate that they were being protected against disease. I did that as a medical student and a resident, and then I was hooked. RSV was going to be a key thing that I would want to be involved with in terms of working into my future. So the evolution of this, I've sort of dealt with the left-hand side of this. There was work going on in terms of developing a purified F protein. A guy whose name you're going to hear later, again, Ed Walsh, basically was the first to identify there was potentially a protein that might work. But everybody was worried, gosh, how do we know we can give this to young babies and we may produce the same exaggerated illness? Whereas live virus doesn't seem to be associated with that. So I and others began working in the live attenuated virus field. But Paul Gleason's work did lead to the potential to have an antibody that could provide sufficient protection, either by taking pooled plasmapheresis sera that had sufficient antibody that could provide protection that got marketed as Respigam, or ultimately a monoclonal antibody, something specifically designed to be selective for RSV. But the problem was this had to be given 5 times intravenously over a season, was really only suitable for those highest risk babies, typically very premature infants to children with young disease. So the overall problem hadn't been solved because most of the hospitalizations were really occurring in children that were older. I'm sorry, that were not prematures, but otherwise healthy infants. And so it was about this time I said, gee, I'd really like to be more expansive in terms of my engagement as far as vaccines are concerned. And this guy, George Sieber, basically approached me about taking on a job. And I will tell you right now that I would not have hired me, but I was very fortunate that he hired me. Why? Because I really didn't have any industry experience. Either he was foolish or he saw something in me that thankfully blossomed. I'm forever grateful. But perhaps as more or equally grateful to the person on the right. Jill Hackle, I actually took her job when I assumed this, and she now reported to me. So here I was, a person with no industry experience, and she was now reporting to me. She was the most gracious person in terms of educating me about what I needed to know in terms of industry. We've remained fast friends to the present. She went on about 7 or 8 years after some remarkable work that we did on developing pneumococcal vaccines to retire from her Pfizer career and become a rabbi. And I was very pleased to be one of the few Gentiles invited to her ordination. And actually, I just saw her again this past spring on a visit to New York. Emilio Amini, for nothing else, it's just fun to say his name, Emilio Amini, an Italian from Little Italy in New York, and I basically came together because he was the successor to George Sieber, and he further built on the foundation that George had built. I think it was his Jesuit upbringing maybe that made us so compatible in terms of the notion of service, but he also remains a lifelong friend. But the foundation that these people built was what allowed us to be able to move forward later to be successful with the vaccine. So then, in about 2013, what's shown on the right side happened. I'm going to tell you a little bit more detail about that. So here I am with Barney, his wife Cynthia, and my wife Gina. Cynthia, an accomplished physician in her own right and psychiatrist. And why are we standing there in my house in New York? It's because Barney was locked out of his office because there was a government shutdown. And many of you know when the government shuts down, people actually can't even go to work. And so he had called me on the phone that day and said, hey, Bill, we're going to be traveling up in your area. Could we drop by? And of course I said, sure, let's come by. I've known Barney all this time. And we would socialize a bit. And I said, when he showed up, I said, by the way, Barney, I saw that in science you had actually managed to crystallize a pre-fusion construct of this F protein that I mentioned before. Were you able to actually, stabilize it further so it's potentially used as a vaccine? He said, well, as a matter of fact, here's all the information I was going to present at the scientific meeting that I just returned from, but I couldn't go. And in fact, he had, with the aid of Jason McClellan, who'd actually recruited from his lab, who'd been frustrated in another lab, developed the pre-fusion structure that essentially enabled the ability to move forward with an RSV vaccine. I said, this is fantastic. I and my future boss, Katherine Jansen, basically went down to visit with him and confirm this result. And that led to basically a better understanding that all the time going back to the 1960s that people had worked on vaccine, we've been working on basically the spent form of the protein, which is shown on the right, instead of the correct form to generate an appropriate antibody response. So if you can visualize this globular structure as what you want to make an appropriate immune response to protect against RSV, it quickly triggers to this sort of elongated molecule that generates very low antibody, whereas if you can manage to stabilize it, which Jason McClellan did in his lab, that allows you to have a huge amount of antibody. And the rest of the story is that we now have, you hear this advertised on the news, a Pfizer vaccine, a GSK vaccine for adults for RSV, and the Pfizer vaccine is now one that is recommended for use in pregnant women to protect their babies. So that in itself would have obviously fulfilled my career in addition to the other opportunities I've had to work with really talented people to develop vaccines. And by the way, ironically, Barney's my house guest this week. He's actually giving a lecture at the Brockman Lecture at the University of Michigan. I have a flyer here I can show you if anybody wants to attend that, if you want to hear directly from the horse's mouth. But there's more to the Barney story. And again, this just gives you a representation of how the virus actually enters the cell. It sort of goes from this globular state to this state, to the more elongated state, and allows the contents of the virus to enter the cell and take over and replicate itself. Okay. So, you may be saying, well, why the heck are you telling me all about RSV? Didn't you come here to tell me about COVID-19? And after all, I think it's worthwhile as we think back with our own time machines back to 2020, how horrific the circumstances were. I think as many of you, particularly given the University of Michigan's proximity, can appreciate, it was particularly challenging for us in the New York area, where people that were part of my team would come in having walked past portable morgues sitting in parking lots because there were so many people dying that they could not manage. So there was a real crisis afoot, and happily now it's mostly in the rear view mirror. But why? So it turns out, and this is something I don't know that anybody talks much about publicly, but is something that I think is worth hearing. The very approach that was used to stabilize the RSV F protein, which in its own right yielded a valuable vaccine, was then used to look at past coronavirus infections, the original SARS you may have heard of, or the original Middle East respiratory syndrome, both of which could have high mortality but didn't transmit as well. Why? Because there was a concern that maybe they would come back. So Barney and his lab had already set to work and stabilize those two viruses. So it became a matter of quickly just pivoting to essentially do the same thing for the SARS coronavirus 2 and stabilize it. So it's a foundational moment that the RSV stabilized protein did this, or that work led to this outcome. And if that had not happened, it's my belief, and I think many other people would share the same, that there would have been millions of more lives lost in the time it would have taken to essentially develop a vaccine. All right, so somehow I've frozen. Uh-oh. Let's see if I can do it down here. There we go. Okay, so this is quite an eye chart, but the purpose is only to show you, if you look over on the right-hand side, there are basically a substitution of 2 proline amino acids for an asparagine and a leucine that made this happen. You see those little elongated structures there around D, and then the globular ones? Looks similar to what we talked about at RSV. Same sort of principle. That globular structure led to significant levels of neutralizing antibody when you inject it into animals, and likely then would be associated with protection. That was for MERS. Kizzmekia Corbett, person from North Carolina who came to study under Barney Graham, became the fundamental person who made that discovery. Meanwhile, on the mRNA front, which became the vehicle by which the antigen would be presented, these two people made remarkable discoveries about how you could essentially engineer the RNA so that the cell would not chew it up too soon and would not develop an immune response too soon that would shut things down before you could develop a vaccine. Catalina Carrico, who, by the way, basically got demoted because RNA was felt to be a backwater for an extended period of time before this discovery was made. Well, turns out that Barney, Kizzmekia, Drew Weissman, and Catalina Carrico all were named Time Heroes of the Year for the work that they did that enabled the COVID-19 vaccine. And Catalina Carrico and Drew Weissman just won the Nobel Prize in medicine in 2023. Now, how does it work? Whoops. It works basically by taking a lipid, putting the mRNA, getting into the cell, allowing the cell to produce the appropriate sort of antigen to produce an immune response. And so with that background, because we at Pfizer had basically already developed a relationship with the two people on the left, and again, time doesn't permit me to tell you, but these two people are two of the most remarkable people in the world, and I'll be happy to answer questions about them for those that can stay. But we basically grouped together and decided, all right, we're already working with these folks on the left for a flu vaccine. We can probably do something to quickly develop a COVID-19 vaccine with them. But it was a daunting challenge. Why? Because the notion was we were going to have to do this from the signing of the agreement to December in 9 months. And we were already 2 months behind the competitor, Moderna. And so in the context of, I'll just tell you right now, I'm pretty competitive. I wanted us to be first on top of also essentially addressing a public health need. And so that was challenging. Why? Because sometimes it takes decades. This is an example of the cold-adapted flu vaccine, which has a Michigan connection. John Masab, who was at the University of Michigan, developed the Arbor strain that ultimately led to flu mist and the cold-adapted vaccine. That was, decades. We measured it in presidents. That was really brainchild of Harry Greenberg, which I thought was clever. Typically, it takes at least 10 years. So credit to this man. Albert Bourla is saying, we're going to do this. He was the head CEO. He himself has a remarkable story. He's lucky to be even on the planet because his mother was literally, as a teenager, lined up against the wall as a Greek Jew before the individuals were shot, machine gunned, and basically pulled from the wall. That's another remarkable story. But in any case, we had to compress this to an incredibly small period of time, which we were successful in doing. And again, I'm happy to tell you more about the details. But it was due to these folks. Alejandro Gertman, You can see all the other names. Alejandra is my actual successor. She actually fled Argentina because of oppression as a Jewish Argentinian and came to the United States, and I recruited her from Mount Sinai in 2005. Nick Kitchen, who led the strategy for our group for the vaccine. John Perez, who dealt with all the data issues. Steve Lockhart, who helped me expand the potential for the vaccine to the rest of the world. Andrea Kawine, who was tending bar and actually preceded me at Pfizer and the legacy Wyeth company, and was taken from the bar to work in the lab and made her way to actually being responsible for the largest part of my organization. Dan Scott, seen here when we were actually trying to move forward. Pneumococcal did so successfully in China. He actually was responsible for kind of maintaining the rest of what we were trying to do, or much of what the rest of what we were trying to do, as well as working with Wendy Watson, shown on the right, who's also a Michigander. Judy Absalon played a critical role in our ability to communicate effectively. He's really responsible for working with the New England Journal of Medicine. And then Ken Curry, who basically was responsible for the statistics and was the best person I know of in terms of being able to give a lay representation of what was happening. So I'm going to quickly go through these. I'm happy to talk about all of these people I've had long-standing relationships with. The picture shown here is on the right-hand side is an actual screenshot of how we were working in a virtual environment, including Rod McKenzie, who I was hopeful David would get a chance to hear as a Scotsman. I'd actually kind of hoped he would be a member of this church because they might have hit it off. Anyway, all of that was happening, and then there was this group, the outside group of the Data Monitoring Committee, who is responsible for managing the safety for the vaccine. And I would just want to give you a point in time, which was the most poignant moment of my entire life, and many of these people have said the same thing. So it was November the 8th, 2020. We had gone through all phase 1, chosen the dose, gotten the right sort of vaccine. Basically, I had recruited over 43,000 people in a very short period of time to receive the vaccine. And in addition to monitoring the safety, their job was to take an early look that we couldn't ourselves look because it would sacrifice the integrity of the trial to determine whether or not we'd been successful. And so on November the 8th, they meet. We're all meeting virtually, and we send them off with these few people listed at the bottom in a firewalled way to look at the data to say whether there's going to be success. And I told them as they were going in, they knew this anyway, that of 7 billion people on the planet, they would be the first 5 to know whether there was hope. And so they went off, and they could have come back with 3 answers. It's futile. That would have been horrific. You're not quite there, but there's still a possibility it could be effective, or it works. And so it's like waiting for a jury. I'm not a lawyer, but I imagine it's like waiting for the jury to come forth. We're sitting there waiting for them to come back on screen. And I'm sitting thinking, waiting, gosh, it's taking a long time. You know, that doesn't sound good, right? They come on the screen, one by one. And you know how it goes with screen types of things. You know, it's technology. It's not all working quite right, and you're kind of waiting for people to come on. So it just prolongs the suspense. So I'm sitting there looking at the screen, trying to read their faces. And I said, did we fail outright? Did we, succeed? I said, well, they don't look morose. So maybe we didn't fail. So then everybody gets on the screen, and the smiles come out, and they said, the vaccine is highly efficacious, very safe. We encourage you right now to call the FDA and essentially move forward to get this approved. And sometimes I break up. I probably will. The reason why it took so long, they were so overwhelmed at the time that they had to recompose themselves about what they just witnessed for the first time. So anyway, it's an extraordinary group of people, all friends and colleagues, many of whom I've known for decades. I'd actually handpicked essentially these folks to be part of our DMC, and it really worked out very well. And so the results. The vaccine proved efficacious, got the emergency use authorization. We published it. There I am presenting to the FDA. I was very grateful to have the opportunity to person for Pfizer that basically represented the data data. And I think you know this man, Arnold Monto, emeritus professor here in the School of Public Health at the University of Michigan, at 88 was chosen to essentially chair the VRPAC committee. The VRPAC committee is an FDA advisory committee. If you've never had a chance, they're public. I highly encourage you to go online, look up VRPAC, VRBPAC, listen to a meeting. You'll, I think, gain additional reassurance about what's done to ensure the safety and efficacy of vaccines. And then subsequent to that, the ACIP met, another committee, same charge, except their charge was, okay, it seems to work. To whom do we recommend it? How do we essentially get it out to the people who need it? And I think if, like you, like me, there was a personal celebration here. In my case, not only was it important because I thought we had achieved something that was really going to be meaningful to protection of millions of people throughout the world, but in my own case, my son John is so disabled that even a minor respiratory illness typically put him in the hospital every year. And as I tell people, I felt like we were living, my wife and I were living on borrowed time. That if he had gotten infected before we managed to develop a vaccine, then I think there's a chance that he would have perished. So I was grateful on both a personal and a global level for this having to been accomplished. Of course, there was a lot of additional work. You know about this, looking at boosters, trying to get into additional populations, figuring out how we're going to deal with variants. And Charu Sabharwal, native Estonian and recently disappointed Astros fan like me, go Tigers, contributed greatly to the ability to move forward this into the pediatric population. And James Baber, a Kiwi in Australia, an ocean swimmer, does a great Freddie Mercury tribute, basically is working on how we would give this with other vaccines, because it's common that you want to take this when you're getting your flu vaccine, for instance. And so a lot of pharmacovigilance done to ensure folks about the safety and the effectiveness of the vaccine. And this is how the rare events of anaphylaxis, a severe allergic reaction, and myocarditis have been seen in a small fraction of, particularly for myocarditis, in a small fraction of young adults and adolescents. But the benefits versus the risk associated with the vaccine still highly outweigh the benefits still highly outweigh the risk, and that's why the CDC continues to recommend the vaccine. So how well did it work? Well, Louis Jodar in our medical affairs group, a Spaniard with, frankly, the passion to match, basically was responsible working with the CDC, working with other entities to show efficacy. This is the efficacy that was first published from the CDC in the first months. You can see all those things shoved to the right, indicating high efficacy, even amongst those that had only received a partial immunization series. So remarkable. And then this just came out. And I'm not getting any royalties or anything for this book. I just happened to hear about it on NPR from Francis Collins, an evangelical Christian who basically headed the Human Genome Project and then went on to lead the NIH during this period of time. And he writes here, and I heard him say this, and then I saw the New York Times article. Future historians will judge the development of safe and effective mRNA vaccines for COVID in 11 months as one of the greatest medical achievements in human history. We felt that at least we were on a path to conquering this disease and stopping the terrible death toll, and to a major extent that came true. Current estimates by the Commonwealth Fund, a nonprofit foundation supporting research on healthcare, are that more than 3 million lives were saved in the United States between December 2020 and November 2022 by COVID vaccines. If you were vaccinated, you might be one of them. I might be also. And I should say that recent data, even last year, despite the fact that most of the population has either been vaccinated or experienced natural infection, still offered a 50% protection rate against disease. And it's believed that when you look at actually the number of hospitalizations, 88% of all hospitalizations are in people that have not been vaccinated. So I encourage vaccination for obvious reasons. So I'm incredibly grateful for all the folks that made this possible. In the interest of time, I'm not going to talk about this. I did want to mention Babalwa Jangalati, who is the woman you see there kind of in the front with the colorful garb. She was responsible for a good deal of the work we were doing in Africa, and I just love saying her name, Babalwa Jangalati. And then this is my group in Asia Pacific and Japan, particularly Kogawara-san, a Shintoist who was very devout, and helped us propel, along with the folks in Australia, our ability to bring this to the rest of the world. So I want to end with one story, and I'm sorry, David, some of you will have to relate this story to him because I wanted him to really get a chance to hear this because I think he'd be taken with it. You may recognize this particular pin, this particularly coming from Ann Arbor, Michigan. So this was a pin given to children who volunteered for the poliovirus Salk vaccine trial that led to the approval of that vaccine and the general use that began to eradicate polio. And Tom Francis was the person who was responsible for that trial right here at the University of Michigan in Ann Arbor. So why am I showing you this? So Ed Walsh, one of the people that I mentioned to you, was one of our investigators early in phase 1. So before we had any evidence that this vaccine was going to work at all. I mean, it seemed to be producing in animals what we wanted to see, but this was the first vaccine ever to use an mRNA technology. And I should have mentioned the reason why was it would be the fastest to getting to a vaccine. So it was sort of taking the bet that it would work because it needed to work for us to be able to have that impact on lives that I was showing you. And so one of the first volunteers that walked into Ed Walsh's suite was wearing one of these polio pioneer pins. So think about that for a moment. Think about the seed that was planted in that 4 to 5 year old child. And here this person was bookending their life, doing something really great for humanity. So, having that happen, I said, we're going to do the same thing. So, working with Judy Sewards, we said, we need to honor all the people that basically participated in these trials. And so, if they were children, they got a teddy bear, if they were, and a pin that you can see it says "In Pursuit of Hope" and makes reference to the trial. And so, I have this fantasy then that sometime in the future, someone will bring in a bedraggled teddy bear or be wearing this pin for some future health emergency where they basically volunteered early in their life and are prepared to do it again. So that pretty much ends my talk, but I'm hopeful I gave you a little bit of a sense of what went behind the scenes, the type of people that came together. Again, a diversity of individuals, Christians, Jews, Muslims, gay, straight, from all over the world that made this happen. And again, it's really up to you. Do you see God conceived as the power sustaining and guiding human destiny in all of this? Thank you. And I realize I've gone over time, but I appreciate those of you that could stay for staying, but I'm happy to hang around if anybody wants to ask questions.
Yeah, just so we, as a way to close this part, but open up for Q&A. This is a small token appreciation that just is a reminder of the light you're putting in the world by what you've done, but also sharing this story, which was just gorgeous. I'm so glad we have it taped so that you can spread it and we can spread it for you. So
yeah, well, thank you very much. Again, if really all those people that you saw, particularly the people who volunteered for the trial, that are the real heroes in this. I told my team, look, I'm probably just a cheerleader here. And I learned once, actually, one of the most the thing that stuck with me when I appointed me to head the viral, the development. A guy comes back, business leader for that, and the job was to the best of their abilities. And that's what I saw myself doing. When we were stymied in place, okay, I'm going to figure out what the team this.
So would anyone, we have a couple minutes, anyone like to ask a question?
Yeah. Barbara.
Okay.
Yes.
I don't think so. Huh. Okay. be on the YouTube channel too, because I'm sure it will be widely watched. I'm curious, Moderna working just there in tandem with you? How?
Yeah.
Tandem, parallel.
I really wanted to tell you about Ugur Sahin and Islam Tureki, and I was afraid that I wouldn't be able to do it in the interest of time. So I don't know a whole lot about what's behind the scenes. I do know that at one point I had tried to recruit the person from Moderna who worked on their clinical development to be part of my organization, and she said, "No, I really want your job." So she went and found something elsewhere, which ended up being at Moderna. But they had been working in the mRNA space for a long time, already working on potentially anti-cancer approaches that are really lend themselves to using mRNA technology. They had already put various vaccines, including influenza vaccines, in the clinic, so they had a big head start. Those of you that haven't heard enough about this, and I think you can probably access the link, or Jane can help you access it. If you read the story that Barney and I were fortunate to be asked to tell for our university about it gets at some of this. So I think, anyway, Ugur Sahin, he's an Alawite, okay, and I don't know much about the Alawite religion. It's sort of like a spin-off, as I understand it, of Islam in a way. A Turk who came to Germany, okay, as an immigrant, and he was in high school, and those of you who know anything about the German educational system, they're very ordered in terms of selecting people early in life, whether they're going to encourage them to go to trade school or they're going to go to a more academic education. His teacher said, "You're going to trade school. You're going to trade school." A neighbor of his interceded and said, "No, you're making a big mistake." So Ugur, basically, while Moderna had its own folks developing an mRNA vaccine, he had already developed monoclonal antibodies with his wife, interestingly enough, Islam Tureki, who thought she was going to be a nun, thankfully for us, basically ended up marrying him. They became a formidable team. That's why they're on that Time magazine cover in terms of developing the vaccine. In fact, I'm told that right after their marriage ceremony, they went back into the lab. They basically first developed intervention strategies using monoclonal antibodies against cancer, but that wasn't enough for them, and they became billionaires. They wanted to do this as well. Ugur doesn't own a car. He rides a bike to work. I've seen, because we've done a lot of stuff virtually, he lives in a very modest apartment. They live the most modest existence. He's the most modest person, he's certainly the most modest billionaire I've ever heard of. And what I really enjoyed hearing about it, so when he came to a celebration that we were having, obviously, about the success, and I got to meet him face to face for the first time, he was telling me about the town where they'd done their work, which is Mainz in Germany, and how poor a town it was that they had not, for years, ever been able to balance their books. And as opposed to the typical sort of language, and I'm as guilty of this as anybody, saying, "How can I avoid taxes?" and essentially maintain more for my heirs. His attitude was different. His attitude, he says, it was just wonderful that the taxation from the money that they got from the COVID-19 vaccine cleared the books for his town overnight. And so, anyway, just I can't say enough about both these scientific intellect as well as just. And to my knowledge, as I said, he's not a Christian; he's an ally. But I think it again shows God's hand in working with everybody to essentially achieve. Anyway, that's a longer answer than you probably wanted, but it gave me the excuse to talk about that.
Thank you. Bill, I bet you would welcome if people want to hover around.
Yeah, sure.
More questions, but I just want to mark that we are at 11:15, which is the end of the Q&A time. Christian, who has to do this, is doing this next week, and who has young children. Usually he leaves at, has to leave at 11:30, right? So I just want to give. You know what? People are kind of spellbound, I would say. And so, do you want to open it for a few more questions? I just want people to feel free to leave if they have to leave.
Feel free to stay.
But
I'm not going anywhere until the room empties.
So, Sid, you want to?
Well, thank you.
Thank you so much. I have two questions.
I worked with John Rousseau.
Did you?
I'm sorry. Public health. I'm sorry. I didn't realize.
That's okay. Go ahead.
And I saw him with one of these cold adapted
Yes,
viruses
yes.
for influenza. And he worked for years.
Oh, yes.
And then when I heard about how fast they developed the RNA vaccine for I think it's wonderful that the science was able to move so fast.
Miraculous and marvelous.
Yeah, it was.
It was, yeah.
Yeah.
No, John was out, again, I did a lot of my early work, part of it was, we'll talk much about this, but with flu
Yeah.
vaccines,
He
with
did
the
some
strains.
very good
When I
work.
was at Vanderbilt, and then ultimately with Wyeth and Pfizer, and then
Oh,
ultimately
yeah.
it became licensed. Some of you may know, it actually now is the first vaccine that you can deliver yourself from home. You just got approved for that, but that's, decades. I'm sorry, go ahead.
That's okay. I have two questions. They're kind of slightly askew from the line you've taken in telling this riveting story. One is, what's the impact, is there any impact of the discourse about vaccine skepticism on the work of the scientists that were working here, we're working on the project? Is that just noise in the background, kind of cultural noise, or does it ever impinge on what you're actually doing? So that was one question. The other is, and I don't know whether this is even a right question to ask, did the work that you do depend on what the scientists who were developing CRISPR were doing all along this period for RNA delivery, or was that just not a part of what was important to your job?
Both are really great questions. I think I'll take the second one first. This thing depends so much on CRISPR technology, but it does depend obviously on understanding of the genome and how
Right.
it engineered. So that's a key piece. And in some ways you can argue that may have come to some extent from CRISPR technology. The first question is an incredible argument. I will tell you that an unfortunate outcome, and I think you all are well aware of this, of the whole COVID-19 experience is the politicization of the nature of vaccines are protected. And I will tell you, you can see this is public information. It's not just individuals. So this is public information, so I'm not revealing anything out of school. But if you go look online, Florida versus Pfizer, Texas versus Pfizer, you'll see public documents from the governments of those entities questioning the safety and efficacy of the vaccine. So that's sort of state-sponsored skepticism. And in that environment, it's a little wonder that in addition to the challenges associated with COVID-19 vaccine, there are challenges for all the routine vaccines. So we've seen a drop in routine immunization of children, which I consider an unfortunate legacy of that anti-vaccine sentiment. I used to show, and I could have put it in here, a picture I'm fond of that comes from the Week magazine, which shows basically kind of a metaphor of people jumping over a cliff like lemmings, wearing signs, I get to choose, blah, blah, blah. That's sort of the unfortunate circumstance that we're in. And actually, this book is at least designed to try to help quell that trend. Again, I'm not here to serve as an apologist or pushing this book, but I think Francis is on to something because he talks about it not just in the context of COVID-19, but the loss of faith in institution, the loss of faith in facts versus opinions, and the blurring of the margins between those two things. Again, I haven't had a chance to, I just started reading, I just came yesterday, I just had a chance to start reading it. This has had an unfortunate impact, and I think we will see in this country not only measles epidemics, but closely following that pertussis epidemics or whooping cough by virtue of the fact that, and that's worldwide. It used to be the case that that anti-vaccine sentiment was not exportable. In developed countries, each country would have sort of its own reasons for skepticism, but most of the low and middle income countries, the developing world, did not have that as an issue, that's changed. It's exported everywhere now. So everywhere is at risk, frankly, because of this cultural change. Now, I'm optimistic that things will come back, but it's going to require a lot of effort to bring things back.
So I think I'm afraid, I think you need to come back and do another session. So please join me in giving this. And again, I really hope you'll join us next week when Dr. Beckler will, he does he's a cranial surgeon who works with children that have clubhouse and gun violence on faces and reconstructing faces, and how his faith has informed his practice as well. So come back.
And again, I'm happy to hang around if anybody has nothing better to do and you want to ask me something. I'm happy to hang around. Oh yeah, thank you.
He was coming in and out.
Yes. Oh, yes, very much so.
We'll see. I'll listen to it.
I didn't know how much more